Hello everyone, welcome back to the channel. You were watching the BioBrainzz YouTube channel, and this lecture series is specially designed for CUET PG 2026 Life Sciences, paper code SCQP17. In today's session, we are starting Unit 2, which is all about chromatin structure and function. In this unit, we will understand how DNA is packed inside the cell, how chromatin is organized, and why this organization is extremely important for gene expression, cell division, and regulation of genetic activities. We will be covering concepts like Euchromatin and Heterochromatin, nucleosomes, chromosome organization in prokaryotes and eukaryotes, centromeres, telomeres, and many more topics that are very important from both exam and conceptual point of view. So, if you are preparing for CUET PG or other life science exams, make sure you are watching this lecture till the end. Let's begin today's session. Now, let us quickly look at the topics that we are going to cover in this unit. First, we will discuss the organization of chromosomes in prokaryotes, how bacterial DNA is arranged inside the nucleoid region, and how it is compact. Next, we will move on to the organization of chromosomes in eukaryotes, where DNA is packed with histone proteins to form chromatin and chromosomes inside the nucleus. After that, we will understand the different types of chromatin, mainly euchromatin and heterochromatin, along with their functional significance. Then, we will study two very important structural regions of chromosomes, the centromere, which is essential for chromosome segregation during cell division, and the telomere, which protects the end of the chromosome. Finally, we will discuss the concept of a gene, including the modern molecular view of what exactly a gene is. All these topics are extremely important from the CUET and other competitive exam point of view. So make sure you follow the entire lecture carefully. Let us start with the why the study of chromatin. First of all, chromatin structure is fundamental to gene regulation. Whether a gene is switched on or off largely depends on how tightly or loosely the DNA is packed inside the nucleus. Secondly, chromatin organization controls DNA packaging and accessibility. Even though the DNA is extremely long, it has to fit inside a tiny nucleus, and at the same time it must remain accessible for transcription, replication, and repair. This balance is achieved through chromatin structure. Another important point, questions from chromatin organization are very frequently asked in CUET PG as well as NET examinations. So this unit is extremely scoring if your concepts are clear. Now, chromatin organization is especially important for some major biological processes. It plays a central role in epigenetics, where chemical modifications on DNA and histones regulate gene expression without changing the DNA sequence. It also crucial for cell division because chromosomes must condense properly during mitosis and meiosis to ensure correct segregation. And finally, chromatin structure maintains genome stability. Improper packing can lead to DNA damage, mutation, and chromosomal abnormalities. So, overall, chromatin organization connects structure with function, and that is why it is such an important topic in molecular biology. Now, let us move ahead and start next discussion, chromosome organization in prokaryotes. In prokaryotic cells like bacteria, the genetic material is usually present as a single circular DNA molecule. This DNA is not enclosed inside a nucleus. Instead, it is located in a region called the nucleoid, which is not surrounded by any nuclear membrane. To fit inside small cell, the DNA is highly compacted by a process known as supercoiling. Along with supercoiling, the chromosome is also associated with special proteins called nucleoid associated proteins or NAPs. These proteins help in blending, looping, and organizing the DNA. Some important nucleoid associated proteins that you should remember for exams are HU, IHF, HNS, and FIS. You can also see in the diagram that apart from the main bacterial chromosome, small circular DNA molecules called plasmids may also be present. These plasmids often carry accessory genes such as antibiotic resistant genes. So, to summarize, prokaryotic chromosomes are circular, located in the nucleoid, lack a nuclear membrane, are supercoiled, and are organized with the help of nucleoid associated proteins. Now, let us look at some important features of prokaryotic chromosomes. First point, prokaryotes usually have a haploid genome, meaning only one copy of their chromosome is present in each cell. Next, their DNA contains very few repetitive sequences when compared to eukaryotic genomes. Another very characteristic feature is that operon organization is common in prokaryotes. Genes involved in the same metabolic pathway are often arranged together and controlled by a single promoter. In prokaryotic cells, transcription and translation are coupled. This means protein synthesis can begin even before the mRNA molecule is completely formed because there is no nuclear membrane separating these processes. Also, prokaryotic genomes show high gene density. Most of the DNA codes for proteins or functional RNAs with very little non-coding regions. You can relate these features to the diagrams shown in the screen, where the nucleoid region contains the circular chromosome along with plasmids and ribosomes distributed throughout the cytoplasm. So, remember, haploid genome, few repetitive sequences, operons common, coupled transcription and translation, and high gene density. These are classic exam-oriented points for prokaryotic chromosomes. Now, let us move to the second major topic, chromosome organization in eukaryotes. In eukaryotic cells, such as plant and animal cells, chromosomes are linear in shape, unlike the circular chromosomes of prokaryotes. These chromosomes are located inside a membrane-bound nucleus, which separates genetic material from the cytoplasm. Here, the DNA is closely associated with special proteins called histones. DNA wraps around histone octamers to form structures known as nucleosomes, and this DNA protein complex is collectively called chromatin. So, in eukaryotes, DNA plus histone proteins together form chromatin. Another important point is that eukaryotic genomes are organized into multiple chromosomes, not just one. For example, the figure on the right side shows different levels of DNA packing from the DNA double helix to nucleosomes, chromatin fibers, and finally condensed metaphase chromosomes. So, remember these core features: linear chromosomes, nuclear localization associated with histones, chromatin formation, and multiple chromosomes because they are very frequently tested in competitive exams. Now, let us understand the levels of DNA packaging in eukaryotes. In other words, how a very long DNA molecule is folded step by step to finally form a visible chromosome. The first and most basic level is the DNA double helix, which has a diameter of about 2 nanometers. Next, this DNA wraps around histone proteins to form nucleosomes. This produces the so-called 10 nanometry nucleosome fiber, also known as the beads on a string structure. After this, the nucleosome fiber further coils to form the 30 nanometry fiber, which provides additional compactation. These fibers are then organized into large loop domains, where sections of chromatin are attached to scaffold proteins. During cell division, especially in metaphase, these loop structures undergo even higher levels of condensation, forming the thick chromatic structure that we observe in dividing cells. The diagram shown here represents this entire hierarchy from the DNA helix to nucleosomes to zigzag fibers, chromatin loops, and finally the fully condensed metaphase chromosome. So remember the sequence clearly for exams: DNA double helix, then nucleosome fiber, then 30 nanometry fiber, then loop domains, then metaphase chromosome. Now we come to one of the most important concepts in this unit, the nucleosome, which is known as the basic structural unit of chromatin. In eukaryotic cells, DNA is wrapped around a group of histone proteins to form a nucleosome. Specifically, the DNA coils around a histone octamer. This histone core is made up of 8 proteins, 2 molecules each of H2A, H2B, H3 and H4. Approximately 146 base pairs of DNA are wrapped around this histone octamer in about 1 and 3/4 turns. Between two neighboring nucleosomes, there is a short stretch of DNA called linker DNA. This linker region is associated with another histone protein known as H1, which helps in further compacting the chromatin fiber. The diagram on the right shows how DNA wraps around histones to form nucleosomes, which then fold to produce chromatin fibers and eventually chromosomes. So, for exams, remember these key points very clearly. Histone octamer composition: about 146 base pairs of DNA per nucleosome, and the role of H1 with linker DNA. Now let us move to the third topic, types of chromatin. Chromatin mainly exists in two major forms, euchromatin and heterochromatin. First, let us talk about euchromatin. Euchromatin is lightly stained when observed under the microscope because the DNA is less tightly packed. It is generally transcriptionally active, meaning that the genes present in euchromatin region are usually being expressed. Structurally, Euchromatin is less condensed and is therefore more accessible to transcription machinery. It is also gene-rich, containing a large number of functional genes. Now coming to the second type, heterochromatin. Heterochromatin appears darkly stained under the microscope. Due to its highly compact structure, it is mostly transcriptionally inactive. So, genes present in these regions are usually not expressed. Heterochromatin is highly condensed and often contains repeat rich DNA sequences. The micrograph shown in the center of the slide highlights the difference between these two types. Lighter region correspond to euchromatin, while the darker patches represent heterochromatin. So remember, light staining and inactive means euchromatin. Dark staining and condensed means heterochromatin. Now, let us further classify heterochromatin into two main types, constitutive heterochromatin and facultative heterochromatin. First, we will discuss constitutive heterochromatin. This type of heterochromatin is always condensed in all cells. It mainly contains satellite DNA, which is highly repetitive in nature. Constitutive heterochromatin is typically found at centromeres and telomeres, where it plays an important structural role in chromosome stability. The image at the bottom shows darkly stained region on chromosomes which usually responded or corresponded to constitutive heterochromatin. Now coming to the second type, facultative heterochromatin. The name suggests the type can switch between condensed and relaxed states depending on developmental stage or cell type. A classic example is the bar body, which represents the inactive X chromosome in female mammals. The micrograph shown on the right highlights a dense spot inside the nucleus, which is the bar body formed due to X chromosome inactivation. So remember, constitutive heterochromatin is permanently condensed and structural, while facultative heterochromatin is reversible and regulatory. Now let us move to the fourth topic, the centromere. The centromere is defined as the primary of a chromosome, that narrow region which appears when chromosomes are highly condensed during cell division. This region serves as the site for cytochrome formation. The cytochrome is a protein complex that assembles on the centromere and is essential for chromosome movement. The centromere is also required for the attachment of spindle fibers during mitosis and meiosis. These spindle fibers pull sister chromatids apart and ensure that each daughter cell receives the correct set of chromosomes. Because of this role, the centromere is extremely important for proper chromosome segregation during both mitosis and meiosis. You can see in the diagram how the centromere divides the chromosome into two arms, the shorter P arm and the longer Q arm, with telomeres present at the end. So remember, cytochrome formation, spindle fiber attachment and accurate segregation. These points define the centromere very well for exam purpose. Now let us classify centromeres based on their position on the chromosome. Depending upon where the centromere is located, chromosomes are grouped into four main types. First is metacentric type. Here, the chromosome is located almost in the middle. So, both chromosome arms, the P arm and Q arm, are nearly equal in length. Second, in the submetacentric type. In this case, the centromere is slightly away from the centre, which result in one arm short and one long arm. Third is acrocentric type. Here, the centromere is located very close to one end of the chromosome, producing a very short P arm and long Q arm. And finally, the telocentric type. In this case, the centromere is present right at the end of one arm is visible. The type is commonly in some organisms but not normally found in humans. The diagrams on the left clearly illustrate these four centromere positions. So, revision, remember, metacentric middle, submetacentric slightly off centre, acrocentric near one end, telocentric at the extreme end. Now, let us move to the fifth topic, the telomere. Telomeres are specially DNA protein structures present at the end of linear chromosomes. They are made up of repeated DNA sequences. In humans, the telomeric repeat sequences is TTAGGG, repeats thousands of times. The main function of telomeres is to protect chromosome ends from degradation during DNA replication and other cellular processes. They also prevent chromosomes from undergoing end-to-end fusion, which could otherwise lead to serious chromosomal abnormalities. In the diagram shown here, you can see telomeres at the both end of the chromosome along with the centromere in the middle and the P and Q arms. So remember, telomeres are repetitive DNA protein structures at chromosome ends that protects the genome and maintain chromosome stability. Now, let us understand the functions of telomeres. First of all, telomeres help in maintaining genome stability. By protecting chromosome ends, they prevent unwanted DNA damage responses and chromosomal rearrangements. Secondly, telomeres plays a role in solving the end replication problem. During DNA replication, the extreme end of linear chromosomes cannot be completely copied by normal DNA polymerase. Telomeric repeats act as a buffer sequences that can be shortened without losing essential genes. The length of telomeres is regulated by a special enzyme called telomerase, which adds new repeat sequences to chromosome ends, especially in germ cells, stem cells, and many cancer cells. Another important point is that telomeres gradually shorten with each cell division, and this shortening is strongly associated with cellular aging. The figures shown here illustrate different aspects forming of the protective T-loop structure, binding of telomerase associated proteins, and the progressive shorten of telomerase over successive cell division. So remember these four key functions: genome stability, end replication problem, telomerase regulation, and aging. Now we come to the final topic of the unit, the concept of a gene. Traditionally, a gene is described as the basic unit of heredity, something that carries information from one generation to the next. In molecular terms, a gene is a segment of DNA that encodes either a protein or a functional RNA molecule such as tRNA or rRNA. A gene is not just the protein coding part, it also includes several important components. First is the promoter, which is the DNA sequence where DNA polymerase binds to begin transcription. Second is the coding region, which actually contains the information for the RNA or protein product. Third are the regulatory sequences, which control when, where, and how strongly a gene is expressed. The diagram on the right shows the complete flow from the DNA with exons and introns to transcription into RNA, splicing to form matured mRNA, and finally translation into a polypeptide chain followed by post-translational modification to form a functional protein. So remember, a gene includes regulatory elements, coding sequences, and promoter regions, and it ultimately produces a functional RNA or protein. With this, we complete Unit 2 on chromatin structure and function. Now, let us look at the modern view of a gene, which is slightly more advanced than the classic definition. Earlier genes were thought to be only the protein coding regions of DNA, but today we understand that a gene is not just the coding sequence. A modern gene includes several regulatory and structural components. It contains enhancer, which increase the level of transcription, and silencers, which reduce or shut down gene expression. It also includes introns and exons. Exons are the sequences that remain in the matured mRNA, while introns are removed during RNA splicing. Another very important concept is that one gene can produce multiple products through a process called alternative splicing. Depending on how the exon are joined, different protein isoforms can be formed from the same gene. The diagram on the right shows distal regulatory elements like enhancers and silencers interacting with promoters through DNA looping, which helps control gene expression. So, in summary, the modern gene concept includes regulatory elements, non-coding regions, and the ability to generate multiple functional products. Now, let us quickly compare prokaryotes and eukaryotes in terms of chromosome and gene organization. On the left side, we have prokaryotes, like bacteria. They usually possess a single chromosome, which is circular in shape. The DNA is located in a region called the nucleoid, which is not surrounded by a nuclear membrane. DNA packaging here mainly involves supercoiling and nucleotide-associated proteins rather than true histones. Chromatin states like Euchromatin and Heterochromatin are absent in prokaryotes. Plasmids are commonly present and they contain accessory genes. Also, prokaryotic genomes contain very little non-coding DNA and genes are often organized into operons. Now, the right side, we have eukaryotes such as plant and animal cells. They contain multiple chromosomes and these chromosomes are linear. The DNA is enclosed inside a membrane-bound nucleus. DNA is wrapped around histone proteins forming nucleosomes and chromatin. Here we clearly see different chromatin states, euchromatin and heterochromatin. Plasmids are rare in eukaryotes. Eukaryotic genomes contain Large amounts of non-coding DNA and genes are usually transcribed individually rather than in operons. The table summarizes these major differences: chromosome number, shape, location, DNA packaging, chromatin organization, plasmids, and non-coding DNA. So for exams, this comparison is extremely important. Questions often come directly such as tables. Let us do a quick revision of Unit 2 and highlight the most important focus areas for your exams. First, nucleosome structure. Remember the histone octamer composition. 2 each of H2A, H2B, H3 and H4. About 146 base pairs of DNA wrapped around it, with H1 associated with the linker DNA. Next, Euchromatin whereas Heterochromatin. Euchromatin is lightly stained and transcriptionally active, while Heterochromatin is darkly stained and highly condensed. Then, Constitutive whereas facultative Heterochromatin. Constitutive Heterochromatin is always condensed and rich in satellite DNA, commonly found at centromeres and telomeres. Facultative heterochromatin can switch states, the classic example being the bar body. After that, centromere types: metacentric, submetacentric, acrocentric and telocentric based on centromere position. Next, telomere sequence and function. In humans, remember the repeat sequence TTAGGG and its role in chromosome protection, genome stability, aging and telomerase regulation. Then prokaryotic whereas eukaryotic chromosome organization, circular whereas linear chromosomes, nucleoid whereas nucleus, absence whereas presence of histones, and operons whereas single gene transcription. Finally, gene definition. Comparing the classic and modern views. Modern genes include regulatory elements like enhancer and silencer. Introns and exons can produce multiple products through alternative splicing. Make sure you revise all these points carefully. They are extremely important for CUET, PG and other competitive exams. With this, we will complete Unit 2 on chromatin structure and function. Before we wrap up today's lecture, let me quickly tell you our student support package, which is completely optional. If you are preparing seriously for CUET PG 2026 in Life Sciences or Plant Biotechnology, you can check the description box below for the registration link. This support package includes Unit 2 detailed PDF notes, previous year question based MCQs, most expected questions for CUET PG 2026, a personalized study schedule, and full revision material with practice sets. For students who need additional guidance, we also provide academic mentoring, doubt clearing session, and CUET focused strategy planning. If you want to join our regular batches, details are mentioned on the slide, along with special student offer for earlier registrants. And if you need any more information, you can directly contact us at the email shown here. All our core lectures are available free on YouTube. You can upgrade only if you want structured mentoring and extra support. Thank you for watching this lecture on chromatin structure and function. Before you go, make sure you subscribe to BioBrainzz for our free lecture series. On the channel, you will get high quality exam oriented preparation for CUET PG Life Sciences completely free on YouTube. New lectures, revision sessions and previous year question discussions are uploaded regularly to help you stay consistent in your preparation. If this video helped you, do like it, share it with your friends and drop your doubts in the comment section. Thanks for watching. See you in the next lecture.